This is the pGLO lab and how it relates to all AP Biology chapters. Unit 1, Chemistry of Life, 1.6, Nucleic Acid DNA. DNA gene codes can be read by all living organisms, therefore they are universal to all organisms. In this lab, the GFP gene is transformed into E. coli bacteria. The genetic makeup of the GFP gene is what makes it glow. It ensures this glowing characteristic in the presence of lactose. Unit 2, Cell Structures and Function, 2.5, Membrane Permeability. Within the bacterial cell is a semi-permeable membrane within the phospholipid bilayer. These phosphate heads face inward while the fatty acid tails face outward. In this lab, the E. coli membrane is semi-permeable, so the heat shock is necessary in order to open up the pores of the membrane to make it permeable for transformed plasmid. Unit 3, Cellular Energetics, 3.1, Enzyme Structure. There is conformational change within the proteins that is due to the interactions of substrates. Therefore, within the lab, the EGFP expression is stopped because a protein bound to an operator will stop the transcription of the gene. The shape of the protein will also be altered because of the presence of lactose. And after this change, RNA polymerase is transcribed, can transcribe the EGFP gene. This is Unit 4, the Cell Communication and Cell Cycle, 4.6, Cell Cycle. Binary fission is the process in which bacteria can asexually reproduce, so they don't need to be around another bacteria. They just can do it on their own. They do this through good conditions, and these good conditions within the lab are provided by the L-B bacteria. Unit 5, Heredity, 5.2, Genetic Diversity. So bacteria increases biodiversity through conjugation, transportation, mutation, transduction, and transformation. So within the lab, the E. coli can be transformed only if the plasmid can get past its membrane. This is also why the heat shock is necessary within the lab. Due to the plasmid being able to successfully pass through the membrane is the reason as to why the bacteria can glow. This is Chapter 6, Gene Expression and Gene Regulation, 6.5, Regulation and Gene Expression. Operons are groups of genes that coordinate regulation. So within the lab, the presence of the lactose operon causes a turn on, therefore allowing the transcription process to take place. This results in the GFP gene successfully being transcribed. So this is also why the E. coli can glow. This is Unit 7, Natural Selection, 7.3, Artificial Selection. So opposed to the natural response of these specific conditions, bioengineering allows for the alteration of E. coli and its phenotype, as well as its ability to survive in the presence of ampicillin. So restriction enzymes and the insertion of EGFP into the plasmid allows for this. Unit 8, Ecology, 8.6, Biodiversity. So within populations, the less diversity means that there's less of a chance of survival if there's a change within the population or in the environment in which the population lives. So in the lab, the lack of diversity in the E. coli bacteria in lane 2 resulted in its poor resilience in response to the presence of ampicillin. Thank you for listening.